Continuing with enzyme inhibition and mechanisms, this lecture will finish out our discussion on enzymes. And we're going to go over some strategies by which enzymes accomplish their mission, right? So turn substrates into products. So not so much the rates involved this time, but what strategy does it employ to accomplish the goal? And then we'll talk extensively about enzyme inhibition and how certain molecules called inhibitors play a role in the effects of speeding up, slowing down, or completely stopping enzyme activity. And so some of the strategies at the bottom here that enzymes employ, and we'll talk about four main ones, are covalent catalysis, and that's where the enzyme itself gets involved in the chemistry. And so a functional group, like an amino group or a carboxy group or an alcohol or whatever group it might be, gets covalently involved in the reaction. Okay, so there's an active site nucleophile, perhaps. So the chemistry that happens in the active site involves amino acids or something from the enzyme itself. Okay, so it forms a covalent bond, at least at some point, with the substrate. This doesn't necessarily mean it's double displacement. It just means that it gets involved in the chemistry. Okay? Another simpler way is general acid-base catalysis. Here, one or more amino acid residues, perhaps there are groups or backbone amines, get involved in the chemistry by acting as acids or bases, just a general acid or base, or a proton donor or a proton acceptor. It's not so involved as, say, covalent catalysis, but they do gain or give away protons, right? So it's a little less involved because it only involves a proton, but it is getting involved in the chemistry and acting as an acid or a base catalyst. Okay? The third type we'll talk about is a metal ion catalysis. Here the enzyme has a cofactor, right? That's a metal ion, and that gets involved in the chemistry, right? It might stabilize some negative charges, such as phosphates, or a carboxylate group, or amino group, but it gets involved in the chemistry by stabilizing some arrangement. And without the metal there, it won't happen. So the metal being a positively charged ion will be an electrophile, right? That can get attacked by certain things, or it can get involved in just holding things at a certain distance apart, right? Where without that metal's specific geometry and specific radius, there's no carbon, nitrogen, or oxygen, or any other part of the protein that could accomplish that. Okay, so the metal is required in metal ion catalysis. Right? And then the fourth one here is probably the simplest of the four, where the enzyme really doesn't truly get involved in the chemistry. It's called catalysis by approximation. And the example I like to use for this is, how do you get a couple 4-year-olds to interact with each other? Right? They normally don't interact with each other. They're running around the room because they've had too much sugar or something like that, right? They're running around the room. How do you get them to interact, or at least say hello to one another? Well, you put a couple chairs on the floor, close together, facing one another, and have them sit in those chairs. And then because they can't get out of the chair, right, not easily because they have to stand up, they actually interact with each other. This is what the enzyme does in the catalysis by approximation strategy. It has an active site that has room for, say, 2 molecules, and when they both sit in their chairs and they're at a spot in the active site, they must face one another. And they're generally facing each other in the right orientation as well. An example of this would be our carbonic anhydrase that we talked about quite a few times now. We have CO2 and water. Those will react with each other if you just have them floating around in the air or in solution. But they have to interact in a certain orientation, right? You have to have the water molecule's oxygen attack the carbonyl carbon in the middle of the CO2. It has to arrange itself to bump into it a certain way. What the enzyme does is facilitate that arrangement. So it just has a spot where they both can bind, and that arranges that oxygen and that carbon such that they can have the attack happen. So it just facilitates the orientation and proximity of the molecules. So it's called catalysis by approximation. Not abbreviation, but it just gets the molecules close together and in the right geometry to each other. Okay, so those are 4 basic strategies that we'll talk about. So what does the enzyme accomplish in acid-base and covalent catalysis. Well, both of those, it physically gets involved in the chemistry, either acting as just a proton donor or acceptor, as acid-base, or making a covalent bond, at least temporarily, with one or more of the substrates. Right? Those tend to enhance the reaction by 10 to 100-fold, which is not a lot. Remember, we have some that go 10 to the 17th faster in that OMP decarboxylase, and some not so much. They're only 1,000-fold faster. Well, this is slower than that. It's because the enzyme has to physically get involved, transfer elements from one part to another, or interact with the substrate. It's much slower than, say, the proximity of two molecules, like the one where they're sitting in the chairs next to each other, right? Our catalysis by approximation could be somewhere in the order of 10 to the 8th or 10 to the 12th times faster, because it doesn't have to move. It has no moving parts. Nothing has to be changed or exchanged. And makes for a much faster reaction, right? And by faster reaction this time, I do mean things physically interacting, not turnovers, but the speed at which the reaction takes place. Not how many or how frequently. I really mean the speed of things moving this time. Okay? So acid-base catalysis, covalent catalysis, and even metal ion catalysis, if it's making and breaking bonds, is much slower than simple catalysis by approximation, because there's no moving parts, right? So the two things that the enzyme accomplishes, no matter which of these strategies it employs, is the two listed here, 1 and 2. There's the proximity effect. I get two things positioned close to one another and in the right orientation or geometry. That's effectively all the catalysis by approximation does. Right? That's its entire strategy. And number 2, the transition state stabilization. We talked about this a couple lectures ago, right? The enzyme's active site doesn't look exactly like the substrate, and doesn't look exactly like the product, or doesn't mimic them after it's induced to. It mimics, or it fits the transition state the best. So it can bend the substrate to that unstable arrangement, or substrates, to their unstable arrangement or proximity. Think I'm tilting the two chairs towards each other to get these 4-year-olds to actually interact, right? So I tilt them or push them in unstable ways to make them interact. So I'm stabilizing that transition state. Whereas without the enzyme, they'd rarely come together and have that type of interaction. Okay, so we're going to consider, how can an enzyme do those things? Well, the shape of the pocket matters, right? The active site has a pocket where things bind, shown at the bottom there. And the shape of that pocket certainly matters. And the things lining that pocket. Are they positively charged? Are they negatively charged? Are they just polar? Are they non-polar? Do they have a certain space for a benzene ring or not? Right? Will it fit a methyl group here? Will it fit a branched carbon here? All those things we need to take into account. Right? So the amino acids that line the pocket, right? Their side chain chemical properties, as well as the shape, the physical shape of the pocket itself. Okay? So if you have an enzymatic reaction versus the same reaction with the enzyme absent, we said it lowered the activation energy. So if you look on the right here, we have our free energy on the y-axis one more time. We could also call this instability, like we did the first time. And on the x-axis, we have our reaction coordinates again. This is just progression, not time per se, but just as the reaction proceeds. In the non-enzymatic or non-catalyzed reaction, we have to go over this huge hump to end up where our products are. Okay? So this number 1 here represents our activation energy or activation barrier without the enzyme present. With the enzyme present, we see E plus S forms an ES complex, right? So that takes a little effort to get it in the active site and then stabilized, right? You see it as a slight hill, and then it goes into a little tiny pit, just a shallow valley there, where ES is bounded. If S is not the right substrate for it, this hill would be much higher and it would not actually bind, right? That would be a poor binding substrate with a high Km. But let's say it's the right one, so it's not so high here. But it also has to push that substrate into its unstable arrangement. So there's another climb. It could have 3 or 4 of these humps. It doesn't make any difference. But the distance it has to climb now is much shorter. You see from E plus S here, where we started, S right here, this dotted line, all the way up to where we started, or end here. I don't mean number 2 here, but if I start with the same point where S is on the y-axis to begin with, and I end up at ES, it's much lower than if I don't have the enzyme present. So I've lowered the activation barrier. However, if your substrate fits the pocket, I don't mean the substrate as in the transition state fitting well. Remember we talked about our stick case. If the magnets look and line exactly like a normal stick, it'll bind to that and do nothing. There's no motivation to bend it. So if it does that, it'll follow this dotted line. It might take an effort to get it in there, but it binds so tightly, so it drops way down on instability, so it's very stable. Now to get up to that transition state is just as high as it was before, if not higher. Right? So it can't climb out of this pit if it binds too tightly to the substrate itself. So it should bind to the ES complex tightly and force the substrate into that unstable arrangement. That's what an enzyme will do. Okay? So it's just another way of looking at our stick case example. Here's showing it during the graphs. Okay? So can we modulate an enzyme's activity? Sure, we already talked about adding more enzyme makes the whole reaction go faster. That's not what I mean here. We've talked about adding more substrate can make it go faster. Again, not what I mean. I mean, yes, those things are true, but outside of that, we've been holding everything else constant. Like we said, we're going to keep the same temperature, we're going to keep the same pH, we're going to keep everything else, the pressure, everything constant. Could altering those other variables have an effect? Let's find out. Do you think altering the temperature could have an effect on the enzyme's activity? Yes or no?
Yes.
And why?
An enzyme might function better at a specific temperature.
So an enzyme might function better at a specific temperature. Why would you think that? Let's make the assumption that if I raise the temperature, it works better. Why would that be a logical assessment?
Could it make the substrates move faster, so there would be more interactions between the enzyme and the substrates?
Exactly. So temperature is not something, I mean, we feel hot and cold and things like that. That's not what we really mean by true temperature. Temperature is the measure of how fast things are roaming around and bumping into each other, right? A higher temperature means things have more kinetic energy and they're moving around. So just like she said, the enzyme is going to run into a substrate more often if they're flying around faster. So yes, temperature should have an effect. Would pH have an effect on enzyme activity? And if so, what might it be and why? Do you think pH could have an effect?
Yeah, it could change their character.
They can affect how stable certain ions are.
Okay, so I didn't hear the first part of what you said.
They can affect how stable certain ions are.
Right, I heard that one, the other person.
Yeah, so it could change their character, whether they are exhibiting positive or negative charge, and that could help them interact or not help them interact.
Okay, so yes, so the ions you're talking about, and whether it has a positive or negative charge, those are ions. You're talking about the amino acids, right? So an enzyme made of amino acids, we're going to ignore the RNA enzymes for a second and look at protein ones. But the amino acids could be protonated, could be deprotonated. They have a certain pKa of those groups, which you've memorized. And a change in the environment, the pH, could have an effect on whether that group is more likely to be protonated or deprotonated or neutral, right? So it could have charges or be neutral, and that could certainly affect what it binds. Your substrate might have a polar group that's looking for a hydrogen bonding partner, and a different pH, the partner won't exist. So definitely pH can have an effect on that. And then finally, we have inhibitory molecules, which we'll spend the most time talking about. And these are molecules that could make the enzyme work less well. It may not necessarily turn it off completely, and they may not bind permanently. They may be only bound temporarily. All right, we'll talk in detail about how those work. But let's summarize your first two real quick, the temperature and the pH, with a few slides. Okay, so here's an example of pH. If I measure my graph one more time, except on this axis, I'm changing the temperature, right, on whatever scale you'd like to put there. And again, looking at enzyme activity or velocity on the y-axis, I notice as I raise the temperature, the enzyme activity tends to increase. And it looks like it's a nice straight line till something weird happens. I fall off a cliff here. What's going on? It looked nice and straight for a while, and then it looks like we fell off a cliff.
That might be getting too hot, so then
Is it denaturation?
Yes. So we're raising the temperature, and like she said, the substrate encounters the enzyme more often. So yes, we're going to get more activity, holding everything else constant. But as we raise the temperature, what's holding our enzyme together? We had hydrogen bonding, we had non-covalent interactions, we had some ionic bonds, we may have had a disulfide bond or two, but the whole thing is being affected by this motion now. The higher temperature causes things to shake. And at some point, it's shaking so much that the thing just unfolds spontaneously. Right? And we're falling off this cliff. So yes, temperature affects activity, and in general, as we raise the temperature, the enzyme has more activity. Up to a certain point, we're going to call that peak here the optimum temperature, right? And it occurs just before we denature the enzyme. Okay? So as an exercise in applying that knowledge, let's look at this cat. Okay, so there's a picture of a cat there. He's some white fur and then some pigmented fur. So the pigmented fur is a result of those cells making a certain pigment, right, like melanin. And there's an enzyme that constructs or uses substrates to make the melanin product. And that enzyme is either making the melanin in the parts of the fur you see as dark, or it's not making the melanin in the parts of the fur that are white. So how can we explain why the cat has certain patterns with temperature? So give me a first thought.
Their like I don't know, chest area is going to be hotter than their external limbs, so it could be that the temperature differences affect the enzymatic activity.
Okay, so let's take that real quick and say the body and the chest and the bulk of the cat, right, is warmer than the feet and the tail. Right? And that's generally true. Right? The feet are going to be colder and the tail is going to be colder than the bulk of the cat. So in the feet and the tail, we're still at a range, a low enough temperature that we have yet to denature that enzyme. And if you get warmer, that enzyme doesn't function, it doesn't make the melanin. It's a good idea. Now I'm going to give you some more evidence that's going to seem to contradict that. That cat's face and its ears are very warm compared to its body, just like in humans, right? Your ears are radiators, right? So they're the warmest parts of the cat, or its face, nose, and mouth, and ears. But it's pigmented again. So does that mean if I raise the temperature even more, the enzyme suddenly works again? That doesn't make any sense. So how can we explain that?
It just works in the reverse that we described.
So as I raise the temperature, the enzyme works worse?
No. I'm saying that the enzyme itself is contributing towards the white, like the non-pigmentation. So the absence of the enzyme might allow for pigmentation.
So the enzyme is what makes the black pigment. So, and white fur is an absence of the pigment. It doesn't make a white pigment. That's not what we're saying. So it seems to contradict our first argument. So how is this possible? So the way we resolve that is there are two enzymes. Right? They do the same chemistry. They make the melanin pigment. However, one's temperature range looks like you see it here, where if it's in the tail and the feet, it's low enough for it to work, right? But if we raise the temperature too high, that one doesn't work. The second enzyme in the lower temperature ranges is extremely low velocity. It doesn't work very well at all. And even when you get to this temperature range, where the red circle here is, or red oval shape here is, it still doesn't work very well. You have to get this enzyme very warm for it to function, right? And you get it warm enough, it'll function in the ears and the nose and so forth. But of course, if you raise it much, much higher, it would also denature. But it generally doesn't. The idea here is there's two separate enzymes that do the same reaction. They just have different temperature ranges in which they operate. One is expressed by certain cells in the cat's head, and one is expressed by cells in the rest of the body. Right? And that's another way to control it, is whether you make the genes for it or not. But even if they made both, you still get the same pattern. And that type of explanation now resolves our why do we see it in very hot and much cooler areas, but not in the areas in between. You need two different enzymes. They do the same chemistry, but they react to temperature in different ways. The range at which they operate is quite different. Okay? It's not the only explanation for the patterning, but it's one explanation. Okay, let's move on to pH. Kind of like temperature, where in temperature we had an optimum, and if we went beyond that, it would denature. Right? And if you went lower than the optimum, it would just give you less activity. It wouldn't denature. Whereas in pH, we also have an optimum. If you look at the graph at the bottom, you see pepsin and chymotrypsin. Pepsin occurs at an optimum somewhere around pH 1.5 to 2, right? Chymotrypsin's optimum somewhere around 8. And if I deviate from the optima, right, the activity drops. If I deviate significantly from this 1.5 or so optimum, the activity falls off so much that it doesn't work at all. We're probably getting into areas where it's completely non-functional, right? Not necessarily denatured, but we're getting into areas where it's non-functional. Chymotrypsin at pH 8 is where it's optimum. If I make it too acidic, it also falls off rapidly and gets to a point where it doesn't work. How is that possible? What's going on if I change the pH there? Pick either one. Pick chymotrypsin. You're at chymotrypsin pH 8. This thing works beautifully, right? It's as fast as it can go. But if I make it a little more acidic, it doesn't work as well. And if I keep raising or lowering the pH, making it more acidic, suddenly down here it doesn't work at all. Say below pH 4, it doesn't work at all. How can we explain that? We're not denaturing it. Anybody have a guess? All right, there's a clue in the shape of the graph. All right, look at the optimum, the point where chymotrypsin's at pH 8, at the very top of the pink curve, and look where it reaches down to near 0, between 4 and 5 pH there. What's the shape of the intermeaning graph? It should remind you of something we've done last time, or 2 lectures ago. We also looked at it during our amino acid lecture. Right, do you notice it's a sigmoidal shape? It doesn't mean cooperativity here. The sigmoidal shape indicates a titration curve. Right? This is exactly what we saw with our amino acids. As we add more base, we saw the pH rise, but then it resisted the change, and then it continued to rise. Only that graph was turned 90 degrees, this one where pH was on the y-axis. So here, going vertical is a resistance to change in pH. So there's something that's acting as a buffer in this region. And where this inflection point is, between the peak and the bottom, is the pKa of that group. So somewhere around pH 6.5 is an amino acid, right, that has a pKa around 6.5 that's acting as a buffer here. Right? So as chymotrypsin is exposed to more acidic conditions, what am I doing to that amino acid? I'm beginning to protonate it. And at some point I overwhelm it and completely protonate it, and the enzyme doesn't work. So there must be an amino acid in chymotrypsin somewhere, most likely in the active site, where it needs to be deprotonated. And if I protonate it, it won't work anymore. Okay, so that's what's going on here. On the other side, if I start at pH 8 and raise the pH up to 9, 10, 11, and so forth, you notice another sigmoidal titration curve. And over there, the pKa is somewhere around 10, right? 9 to 10. And there, I'm deprotonating some amino acid, or one or more amino acids, that have a pKa around 10. And if I deprotonate it, it no longer works. So there's an amino acid that also needs to be protonated, probably somewhere in the active site, for this enzyme to work. So these are pH rate profiles, right? So I can figure out what those amino acids might be by their pKa's. So the one that has a pKa around 10 might be what amino acid? There's a couple I can think of. You can think of 2, I'm sure. And then the one that has a pKa around 6.5 to 7, probably think of one there too that has a pKa around that number. It's not going to be the exact number because remember, microenvironments can shift it a little. But I'm thinking the one up here could be a tyrosine or a lysine, and then the one where it's being more acidic and titrating it might be a histidine, right? Because around pH 6 or pKa of 6. Same thing can be said for pepsin, right? Pepsin, if you look at it, its peak is around 1.5 to 2, and then as we make it more basic, there's a titration curve, the black line, and it has a pKa somewhere around 3, where the inflection point is. So I'm thinking there's a carboxylic acid there that needs to be protonated. And as I raise the pH, it's going to get deprotonated and not function. So I'm thinking there's an aspartic acid or a glutamic acid or possibly even the carboxy terminus of the protein. Right? There's a carboxyl group most likely with this pKa. That's how we read these. So pH does affect enzymes, and that we get this pH rate profile, but it's convenient. It doesn't just fall off. We can find the optimum. We can also have an indication of what amino acids might be involved by the inflection point or their pKa's. Right? And then the question I said, 2 human proteases, where are these 2 enzymes found? Where are they active in the body? Well, pepsin is found in the stomach, which has a pH somewhere around 1 to 2, and that's where pepsin's most active, at pH 1 to 2. It evolved to be active in that environment. Chymotrypsin would not be active there at all. It'd actually fall apart, but it's not found there. Chymotrypsin you're going to find in the small intestine, right? So it's most active around pH 8, the pH of your small intestine. Pepsin, however, is in the stomach, and it gets transferred with the chyme, the food, into the small intestine, and the pepsin molecules just become more food. Because at that point, they're not active anymore, and we digest those too and recycle them. Okay. So here's another graph showing exactly what I was talking about. The graph at the top right says pH on the x-axis, and this happens to be like chymotrypsin here, where we have a optimum around pH 8. And if I deviate from that optimum, if I go more basic, I'm protonating something, right? Deprotonating something, likely an amino group. And if I'm protonating, going more acidic, it's probably going to be a carboxyl group or a histidine or something like that. So you gotta look at where the inflection points are and judge what amino acids might have a pKa near this point. And so, do we really regulate enzymes by changing the pH? I mean, most cells don't drastically change their pH like this. Yes, between the stomach and the small intestine, that makes sense. But in the blood and in the cells, we generally don't vary the pH that drastically. So do we alter enzyme activity by changing pH? In fact, yes, we do, just not that drastic. It's much more subtle. One enzyme we're going to talk about in metabolism when we get to glycolysis is called phosphofructokinase, and it's the third step in glycolysis, we'll learn later. But slight changes in pH cause it to change its activity. And it changes activity by the subunits of PFK coming apart. It's a multi-subunit enzyme. It's got quaternary structure, and all of them have to be together for it to function. But if I change the pH just a little bit, right, make it slightly more acidic, those subunits no longer associate, and then the enzyme won't work. So why does the pH get slightly more acidic? It's because there might be a little more lactic acid being produced by fermentation. If fermentation is going on, we make a little more lactic acid, the pH will tend to become slightly more acidic. Instead of 7.3, it might be 7.2 or 7.15 or something. It's not going to be that wild, but it'll get slightly more acidic, and that'll cause this PFK enzyme to stop working. And if we stop PFK from working, we slow down glycolysis, which will limit the production of more lactic acid. So it limits the harm it's doing. Okay, that's just one example of how pH can be used for that. Another example is the dissociation of oxygen from hemoglobin. Right? As the pH changes, the hemoglobin has a changing affinity for its oxygen. Although hemoglobin is not officially an enzyme, but it can bind and release its substrate at different pHs by different amounts. So same idea. Question? Okay, so on to the last part of the things that can change enzyme activity, and these are going to be our inhibitors. Right? And I put up 2 articles. The one was written by Dr. Waldrop in 2009, and he and I put that paper together. I drew all the silly figures, and he wrote the wonderfully written text for it. And then I followed it up by one in 2015 to take what we wrote in 2009 as a qualitative assessment of enzyme inhibition, and then put some numbers to it quantitatively. But I suggest you read the qualitative one first, or the other one will not make much sense to you. So read the 2009 article first. It's not very long at all. And then read the 2015 one, which is even shorter. I think it's 1 or 2 pages. But read them in that order. Understand it qualitatively first, then quantitatively afterwards. Okay? And I'm going to do the same thing with you here in class with the PowerPoint. Okay, so the first thing we need to do is go over the 2 broad types of inhibitors. Okay, there's the reversible type and the irreversible type. The reversible type is a molecule that when it binds to the enzyme, it's not permanently bound. It can dissociate, it can come off. But while it's bound to the enzyme, that enzyme cannot function. And so it's a temporary shutdown of the enzyme. Don't think of it as permanent. Right? So if a reversible inhibitor is bound to an enzyme, that enzyme at the moment cannot do its job. But that enzyme is not permanently lost. The reversible inhibitor could dissociate, and the enzyme will work as if nothing was ever wrong with it. It'll happen, do its job all over again. Right? As opposed to irreversible inhibitors. Again, like reversible, if they're bound, the enzyme doesn't work, but they never come off. Once they're bound, they're stuck on forever. Okay? So we call those affinity labels. We use them as affinity labels. We call them suicide inhibitors. They're generally very specific about what they bind to. And the best way to stay on and not come off is to make a covalent bond. Okay? There are some that are technically reversible inhibitors, right? Like our pyrrole we talked about last time, our proline isomerase inhibitor, that binds so tightly as transition state analogs that they look like they're irreversible. And we kind of group them with the irreversibles because they're effectively never coming off. Right? But technically they are not irreversible, they're reversible, but we're going to throw them in with the irreversibles because they effectively never come off. So I want to not make a broad statement about irreversibles having a covalent bond, but a lot of them do. Right? Not all of them do, but a lot of them have a covalent bond, and that's a great way to never come off. But it is true that the reversibles generally do not make covalent bonds because they can come off. They just bind through hydrophobic interactions and hydrogen bonding and ionic interactions and all those types we talked about before, even pi stacking and stuff like that. But reversibles do not make covalent bonds, whereas irreversibles generally do. Okay, so of the 2, the irreversibles are kind of the boring group, right? They bind, they never come off, that enzyme will never work again. But along the same lines, that inhibitor will never inhibit again. It's a suicide inhibitor, it's consumed in the process. So if you have a molecule that's an irreversible inhibitor and it binds to its target enzyme, yes, it'll do its job. That enzyme will never work or never function again, but that inhibitor will never bind another enzyme. Right? It's consumed in that process. Whereas with reversible, it's a trade-off. If I bind to my target enzyme and I'm a reversible inhibitor, right now that inhibitor is inhibiting the enzyme. It can't function. But I will eventually dissociate. And that enzyme will work again, but I can go and bind it again if I need to, or another copy of it. So I'm reusable. So there's a trade-off to being irreversible versus reversible. So, and that's shown in the way we prescribe certain drugs. So if you're prescribing an irreversible inhibitor, you're going to need more of it. It's going to be used up quickly, right? It inhibits very well. You're going to have a small dose, and it'll inhibit very well, but it will be cleared quickly, right? You'll need more of it. Whereas reversibles, you're going to need a larger dose, but not as many doses, right? Because it stays around longer. So it depends on the toxicity of the drug too, which one you want to design. So there's a trade-off for using one versus the other. For our discussion today, irreversibles are rather simple. They bind, the enzyme doesn't work, the velocity will be 0. Very simple. This doesn't work anymore. For reversibles, since they bind and release rather quickly, we can have the thing still work, and from our point of view, we see it working more slowly. And again, by slowly this time, I mean less frequently. Right? The enzyme doesn't make product as quickly in the presence of the reversible inhibitor. right? And it doesn't make product at all in the presence of the irreversible inhibitor. Okay? All right, so I apologize for the names. They came up with these names before any of us were born. So we didn't get a say in these names, and we'd love to change them, but I don't think they're ever going to change them. So the three types of reversible inhibition are called competitive, uncompetitive, and non-competitive. And I will tell you, this is one of my pet peeves and my soapbox topic, right? Because they've been repeating the same oversimplified way of saying this in textbooks, biochem textbooks, for the past 3-4 decades, and they still make the same mistake. They oversimplify it and they paint themselves into a corner, and it's wrong, right? So we're gonna unravel that mistake and state this in a simpler, easier to understand way, and it'll make much more sense than the hand waving done to explain these in textbooks. Okay, so tell me what you know about these three types, and we'll clear up any misconceptions. And you may know something that's absolutely right, and it's okay to be wrong here again. We're going to learn from this. So tell me what you know about these three so far.
Competitive inhibitors resemble like what they're blocking, I think.
So competitive inhibitors resemble the substrate. Is that what you're saying?
Right.
Okay. And so you're not absolutely right, and you're not absolutely wrong. Competitive inhibitors often resemble the substrate, I agree, but they don't have to. Right? Now, not everybody will agree with that. They say all competitive inhibitors resemble the substrate. That's not true. Okay? Do they often resemble the substrate? Yes, and in fact, most of them do. But they don't have to. So if you're planning to take the MCAT or DAT or PCAT or any of those, they love asking that question, and that's the answer they want, the one you just said. But it's not entirely accurate. So get the question right, but know you know better. Okay, so that's one misconception we need to clear up. Competitive inhibitors often resemble the substrate they're competing with, but they don't have to. Anything else? There's usually one more. That's a common misconception.
Do competitive inhibitors block the active site so that the substrate attached to the enzyme or interact with it?
I like the way you said that until you said the very last thing. So do they block the active site? Yes, I agree with the verbs you used. Right? Do they bind in the active site? They often do, but they don't have to again. But when you say they block the active site, if you mean physically block it, I don't agree with that. They don't have to. But if you say they block the substrate from binding, however they accomplish that, then I absolutely agree with you. So all a competitive inhibitor needs to do to be labeled competitive is upon binding, the substrate cannot. Right? Can it do that by binding in the active site? Absolutely. Is that the only way to do it? No. Okay? So we'll keep that in mind. You notice a lot of these have a theme to it. It's been oversimplified by making these statements that aren't necessarily always true. That's what I'm trying to get out. Okay? What about, so that's the competitive ones I usually have misconceptions about. What about uncompetitive? What do you know about that one? Anything stand out about that one that you remember? In fact, that one's not even covered for most of these aptitude tests or entrance exams because they run out of ways to explain it. They generally cover competitive and non-competitive, and they don't even call it non-competitive. They call it mixed. But for uncompetitive, it is often said that the inhibitor double parks the substrate onto the active site. Like the substrate, of course, binds in the active site. That's the definition of where it binds. And then the uncompetitive inhibitor, they say, parks on top of it, prevents it from leaving. That's not necessarily true. The prevents it from leaving part is true, but where it binds is, again, irrelevant. As long as when it binds, it prevents the substrate from leaving. By binding on top of it, preventing it from leaving physically, that's an excellent way to do that. But again, it is not the only way to do that. And this is where I have a problem with all these figures in the book that draw it that way, misleading you to believe it's the only way. Okay? And then non-competitive, they love to equate with allosteric, which is not true. We said that at the end of our last lecture. So non-competitive does not mean allosteric. They like to say it binds somewhere other than the active site, and that's not true at all. Right? Non-competitive is simply a sum of the first two. Both options are available. Okay? And we'll talk about what those options are in just a second. Okay, so let's go through those, and I have my favorite analogy of all time to help explain how these work. Okay, so let's talk about competitive first and what I mean by a competitive inhibitor. So I got some verbiage at the bottom down there to help you explain it. I'm not going to read it to you too much. I'm just going to explain it to you. So let's look at the scheme, the kinetic scheme on the top left. Okay, and I'm going to start out pointing the horizontal part first, which you're already familiar with. We have an enzyme E binding its substrate S. E plus S can bind one another and make an ES complex. This is not unfamiliar to you at this point. That occurs at the rate K1. It could fall apart at the rate K minus 1 and so forth. ES can also make product. So it turns into EP and then releases the product to give E plus P. And we said that rate was called K2 or Kcat. Okay? This is all old stuff. We saw this last time. Okay? The new part of this feature, or this kinetic scheme here, is the inhibitor I is able to bind to E, right? Never to ES, only to E. So the inhibitor I can bind to the free enzyme, we call it, and form an EI complex, much like E binding to S forms an ES complex. But if the substrate is already bound, in other words, ES, the inhibitor cannot bind. So this says a competitive inhibitor can only bind to the enzyme if the substrate is not bound. It's true competition. Either S binds to E or I binds to E, never both. If one's bound, like EI, the substrate cannot bind. What must happen first? E and I must dissociate, right? And that dissociation here, that's the up arrow here of EI falling apart into E plus I, is given a rate. It's called KIS. Okay? Much like this was called K1, this is called KIS. It's a dissociation constant. It's the rate at which EI falls apart. Okay? And it must be written on the up arrow, on the dissociation arrow. I've seen textbooks that draw it on the down arrow, which is absolutely wrong. Okay? And I've seen one where they draw it one up and one arrow on the same page. So be careful how you look at those. Right? But what's clear here is the inhibitor I does not bind to ES, never will. Okay? If it does, it's not a competitive inhibitor. So this is the definition of competitive. I only binds to E, never to ES. Okay? Let's look at the effects of that. Okay, we're going to look at two effects. One is what happens to Vmax. Okay? So without the inhibitor around, we're going to call that Vmax. And that would occur, pretend all the stuff about I here is not shown on the page, just ignore it. We just have this horizontal thing. E plus S forms ES. It could break apart into E plus S, or it could form product and release it. Our normal scheme. In the absence of any enzyme, when will Vmax occur? Vmax occurs under what conditions? You're going to hear me go through this algorithm with you 100 times. Vmax occurs under what conditions?
Constant enzyme concentration and saturated substrate.
Okay, so we're going to hold the enzyme constant. Yes, we're going to say that's constant forever now. But the important part there was Vmax occurs at saturating amounts of S. And how much saturating, what do we mean by that? How much S is there? Infinite amount of S, right? As much as we can throw at it. Theoretically, infinite amount, right? Not really can do it in reality, but we'll think about it abstractly. So if you have an infinite amount of substrate around, which you're absolutely right, what will happen to this equilibrium I'm pointing to? So it's E plus S on the left, and you have ES on the right, and it's connected by those alternating arrows. What will happen to that equilibrium if I disturb it by adding infinite amount of S? What will Le Chatelier's say? We're going to do this entire analysis by Le Chatelier's principle. So I add an infinite amount of S. What happens to the E plus S to ES equilibrium? Will it shift?
Shifts to ES.
How far to ES?
It'll just keep on going.
Completely, right? We're adding an infinite amount of stuff to the left-hand side of that equilibrium, right? Can you imagine that? It'll never be able to restore equilibrium. It's an infinite amount. So no matter what it does, it's going to use as much S as it can to try to restore equilibrium. Effectively, there will be no free enzyme around, right? Every E will have an S bound to it. That's what we're trying to say. So then the only thing around would be ES, and of course lots of S left over, but there'll be no free enzyme. And then of course that's the maximum amount of ES, and we know that ES concentration times Kcat is the rate, and that's as fast as it can go. That's Vmax. Under those conditions, would the inhibitor I have anything to bind to? Our competitive inhibitor I, will it have anything it could bind to under those conditions?
Maybe if it gets lucky.
What's the only thing I binds to for a competitive inhibitor I?
Just
E.
the enzyme.
Free enzyme, free E. It'll never bind to anything else, never bind to ES. So under conditions of infinite substrate, will the inhibitor have anything to bind to?
No.
No. You compete with infinity, you lose. Right? So I'll give you an analogy for that. You have assigned seats in the classroom, right? You go and sit down. And at some point during class, I tell you, everybody get up and walk in the hall. Everybody get out of the classroom. And then I let you back in the room at the same time I let an infinite number of toddlers into the room. Will you get your seat? No, of course not. You will lose if you compete with infinity every time. That's the purpose of infinity. So here, under conditions of infinite substrate, that's the conditions leading to Vmax, the inhibitor has nothing to do. So do you think it will affect the Vmax? No matter how much I throw in there, no matter how many students I throw in there, you're going to lose to the infinite toddlers. Right? So under conditions of Vmax, or infinite substrate, when you get Vmax, the inhibitor, the competitive inhibitor, can have no effect. It'll lose. So that's why we say the Vmax is unaffected. Right? With or without the inhibitor, it will not change. Okay? What about Km? So let's go through another algorithm. Km represents the what? What's its interpretation? What does Km represent?
The amount of S needed for Vmax, or 1/2 Vmax.
So that's the definition. It's the amount of substrate required for the velocity to be 1/2 Vmax. But what is its interpretation? What does Km mean to you?
It's the affinity.
It's
So if you have
it's related.
the affinity,
It's related to the affinity. It's not equal to it. How is it related to it?
if you have a smaller Km value, then it means it has a higher affinity.
So reciprocally related, exactly.
Yeah.
So a large Km means weak affinity. A small Km means high affinity, right? So, but in our scheme up here, if you look at our E plus S to ES, with the K1 on the arrow, and then ES falling apart with the to E plus S with the K minus 1, not with infinite amount of S anymore, we're not talking about that anymore, that was Vmax, but under normal amounts of S. Won't that ratio of E plus S and ES come to some equilibrium? Right? It's going to bind and release until we come to some equilibrium. It happens very rapidly. Right? So we're at a steady state at that point. Right? So we have E plus S pool and we have an ES pool. Occasionally we make a product. But the E plus S and the ES are in an equilibrium with each other. Right? Very rapid, it comes to equilibrium. Now I throw in this inhibitor. Right? What is it going to do to that equilibrium, to that binding equilibrium? Will it disturb it? So look at the scheme. We have E plus S forming ES and ES falling apart into the two components, and it comes to equilibrium rapidly. And now I throw in the enzyme. I throw in the inhibitor. The inhibitor is going to bind to the free enzyme. There is some available now. We're not at Vmax. And it'll form an EI complex. So could you imagine it's involving some things on the left in a side reaction? It's pulling some of my substrate away. That's a bad choice of words. It's pulling some of my reactant E away. So I'm removing things from the left-hand side, right? Can you see that? Mm-hmm. And Le Chatelier says if I remove something from the left-hand side, what does the reaction tend to do to restore equilibrium?
Shift back to the left.
I'm going to shift to the left. So the binding equilibrium will shift to the left. In other words, there'll be a little more ES falling apart. Does that make sense? So from our point of view, does it look like ES is falling apart more often than it did before without the inhibitor there? Yeah, because I'm shifting to the left to try to restore that equilibrium. So from our point of view, ES is falling apart more than it was before. And ES tends to fall apart more. It looks like it's not binding as well. So what will the Km appear to do? The true Km hasn't changed, but what will the Km appear to do?
Appear to increase?
It'll appear to be increased because it has a weaker apparent affinity. The true affinities don't change. E and S have an affinity for one another. But in the presence of the inhibitor, it appears to the outside observer that the Km is increased a bit, that they don't like each other as much. It just appears that way. So we have an apparent increased Km. Do I want you to memorize that? No. I want you to be able to derive it in just the way we did. Right? So looking at how it interacts. So in that idea, I want you to apply what we just did to uncompetitive. Okay, we'll get to the graphs in a minute. But apply what we just did for Vmax and Km to an uncompetitive inhibitor. So let's walk through it together. So here's our scheme one more time. I left out all the K's at the top this time because you're familiar with them. But we have E plus S forming ES. That could fall apart into E plus S. Or ES could form product and release it. That's the arrow to the right. In the scenario for an uncompetitive inhibitor, it only binds to the ES complex, never to free enzyme. So competitive and uncompetitive are mutually exclusive things, right? One only binds E, that's competitive, free enzyme. One only binds ES, that's uncompetitive. They bind mutually exclusive things, okay? What effects might this have? Let's do Vmax first, okay? Vmax occurs under what conditions?
Constant enzyme presence and
not
saturation of a substrate.
Yeah, so not so much constant enzyme, we're going to assume that forever, but saturating amounts of substrate, you're right. So under saturating or infinite amounts of substrate, where does that equilibrium between E plus S and ES lie?
ES.
How far to ES?
All the way.
Completely, meaning there'll be what?
Only ES.
Only ES and
Plus B. Product as well.
no free enzyme, right? Every enzyme will have a substrate on it. That's what infinite amount of substrate means. Can our uncompetitive inhibitor bind in that scenario?
Yes.
Yeah, that's exactly what it binds to. It loves that scenario, right? So will the amount of product we make be diminished by the inhibitor's presence?
Yes.
Yes, because you see it's removing some ES from the equation. So unlike competitive, under saturating conditions, the Vmax will still have a change. It'll appear to decrease because of the presence of the inhibitor and because it can bind to the ES. Okay? Moving on, let's do Km. Km represents the binding of ES to E to S to form ES, and it's falling apart, right? Remember, the high Km represents a weak affinity. Low Km means high affinity, the true affinity. Now you throw in the inhibitor. What will it do to the binding equilibrium of E plus S and ES? How will it affect that equilibrium?
Increase ES because you're losing ES.
Right, it's going to involve ES in a side reaction to form ESI, right? So it appears the right side of the equilibrium of removing something. So Le Chatelier says it will shift right to make more ES from E plus S. So to the outside observer, again, the presence of this uncompetitive inhibitor appears to shift the binding towards the bound state, the ES complex. So it looks like they like each other a little better. The true affinity hasn't changed, but our point of view, it appears that they're binding a little tighter due to this inhibitor. So what would that do to the appearance of the KM?
It'll make
And
the
increase
KM value go lower.
It'll make the KM appear to be lower. Exactly. So we call the apparent KM is decreased.
Would
Now-
it be wrong to say that the KM that it appears to have a higher affinity, because ES is being used for ESI, so it can dissociate into E plus P?
So yeah, we're only, when you talk about KM, we don't really talk about E and P. We're talking about the binding of E plus S to form ES and their dissociation into E plus S. Generally because the rate of formation of product is much slower than the rate of dissociation into E plus S. In other words, K cat is much smaller than K minus 1.
Okay.
So the rate of bind and release is far more rapid than forming product. Right? If it's not, then this analysis also has to keep that other thing you just mentioned in their mind. But we're going to assume for a minute that the formation of product is much, much slower, orders of magnitude slower, than the bind and release of substrate. But you're not wrong, but we're going to keep our discussion to the beginning to make it simpler for you. And so in the uncompetitive case, the binding of inhibitor to ES causes the binding equilibrium to look like it's shifting towards the bound state, so it appears to be binding more tightly. It truly isn't, just appears to be. And so our KM appears to be lower. Okay? And in fact, the proportion by which we decreased Vmax apparently, and the proportion by which we decreased Km apparently is the same number. Right? They're both decreased by the same proportion. Okay? And then lastly, non-competitive. A lot of books will call this a mixed inhibition. I'm not sure what they're mixing, but and they call the special case of that a non-competitive. But we're going to call anything where the inhibitor has the ability to bind to free enzyme or ES complex, not one or the other, but it could bind to either one. It's a mixture of the first two. We're going to call it non-competitive. Okay? So let's analyze this one. Would Vmax appear to change? Again, Vmax occurs under what conditions? Go through the algorithm.
Infinite amount of substrate.
Yep, keep going. Infinite amount of substrate. What effect does that have on the binding equilibrium?
There's no free enzymes.
Right, shifts it completely to ES. There'll be no free enzyme available. Can the non-competitive inhibitor still bind?
Yes,
Why?
because it can bind to the ES complex.
Right, it also still has the ability to bind to ES. It could bind to free enzyme if there was some around, but under this scenario there isn't. But it still can bind to ES, just like our uncompetitive can. So will it apparently affect the Vmax? So will Vmax apparent change? So it still has the ability to bind to ES. So even under saturating conditions, it still will have an effect, just like
So
uncompetitive.
if it does the same thing as the last one, then it would.
If it can bind to ES, then it will have that effect.
Right.
So yes, it still has an effect. For the same reason uncompetitive did. It can bind to ES complex. And for the very reason competitive did not. It could not bind to ES complex. Because that's the only thing in this entire scenario that can make product, is an ES complex. If you can alter the concentration of that, you can alter the rate of product production. Okay? So the question here, though, is about Km. What happens now? So we have our normal equilibrium of E plus S to ES, and if I add the inhibitor, does it alter that equilibrium? Go through the same analysis we just did for the other two. So with the competitive, remember the inhibitor bound to E, depleting some things from the left-hand side, so it shifted left to compensate. For uncompetitive, it bound to ES, depleting something from the right-hand side, so it shifted right to compensate. What about here? So it can bind to either E or ES. So at first glance, you might think it doesn't alter the equilibrium at all because it can bind on either side. And that would be true, and that's what a lot of the books call non-competitive. That would be true if I, the molecule I, the inhibitor molecule, has the same affinity for two different things, E and ES. Those are not the same molecules. If I has the identical affinity for E as it does for ES, then no, the Km would not appear to change. But it's very unlikely that one molecule has the exact same affinity for two different things. It usually has a preference. So whichever it has a preference for, E or ES, it could alter it such that the Km appears to go down, or it could appear to go up, depending on the I's preference. So Km could go up or appear to go up. It could appear to go down. It could, very rarely, but it could stay exactly the same. So what the books are calling true non-competitive, or whatever wording they use, is when that assumption of equal affinities is there, which is very rare, it's almost never observed. Or they call it mixed when it's not equal. Right? But I'm going to call it non-competitive. In any case, they just have different affinities. Okay, so to summarize, here's a chart from one of those books where I've blocked out the last part that's misquoted. I don't want you to memorize these effects. I want you to know how to arrive at these answers. And it's all the Chatelier's principle. Figure out what happens to Vmax. Vmax occurs under what conditions? How does that affect the binding equilibrium? Can the inhibitor still bind? Yes or no? How does it affect Km? How does the inhibitor bind and adjust the binding equilibrium? To the left, that means it appears to be weaker binding, higher Km. To the right, it appears to be a higher interaction or a stronger interaction, so a lower apparent Km. Be able to do that for these things. And for non-competitive, it could go either way for that, depending on the relative strengths. Okay? So what I like to do is summarize everything we just said in 2 sentences. Right? That sounds crazy. Can you do that? Sure. I want you to come to the same conclusion I will. Okay? So here, let's look at the one on the right first. So on the right, we have our 3 schemes, once again, that you're familiar with. We have E plus S binding to make ES. ES could fall apart into E plus S, or it could turn into product and release the product E plus P. And that's listed in all 3 cases. For competitive at the top, we can have the inhibitor, I, can bind only to E, forming an EI complex. And it can fall apart back to E plus I because it's not irreversible. It's a reversible inhibitor. It's not permanent. And it does so at a rate called KIS. Okay? For the uncompetitive, we have the same scenario, except it binds only to ES. And at the bottom, our non-competitive, the inhibitor can bind to either E or ES, and they can fall apart at their relative rates, KIS and KII. Okay? I want you to look at the graphs on the left now. And these are a bunch of straight lines. What's the only thing you can tell me about a straight line? It only has 2 features. What 2 things I need to know to make a straight line?
Slope and the y-intercept.
The slope and the y-intercept. The x-intercept is simply a calculation from the previous 2. That's all it is. So we can ignore x-intercept. Don't focus on it at all. I know they like to do that in the books too. So, but you don't see the x-intercept in the equation unless you solve for y equals 0. But let's look at the slope and the y-intercept. Okay? If you remember back from earlier, our last lecture, the slope of this line, these are Lineweaver-Burke plots. It's 1 over v versus 1 over s. The slope of the line is what? What was the slope of this line? It was in our equation that you flipped over and solved for. It was that first fraction of the 2.
It's a Km over Vmax.
Exactly. So it's a pair of constants. Km divided by Vmax is the slope of that line. And then the y-intercept was equal to what? The b in the equation.
Yeah, the inverse of Vmax. So
1 over Vmax, exactly. So the slope of each of these lines is represented by Km over Vmax. And the intercept in all these lines where they meet the y-axis is 1 over Vmax. Now, if all the lines were to lie right on top of each other and be the same line, wouldn't they all have the same slopes and intercepts? That's the definition of the same lines. And then they'd all have the same Vmaxes and Kms. And there would be no inhibition. That would be the definition if all the lines laid right on top of each other. But clearly they don't do that. So in the first case, very simple analysis, look at the first case, case A up there, the competitive inhibitor. Those 4 lines I have drawn share something in common and something not in common. What can you tell me about those 4 lines using that type of description?
They have the same B value.
Same y-intercept with different slope.
Exactly. They have different slopes, but the same y-intercept. Okay. What about in B for the uncompetitive case?
Same slope, different y-intercepts.
Right. The lines are parallel, which you know it means they have the same slope, but they certainly have different intercepts. They cross the y-axis at different points. So it's just the opposite of the before case. That should sound familiar, that competitive and uncompetitive seem to be opposites of each other. And then in the last case, what can you tell me for non-competitive? We're talking only about their slopes and y-intercepts.
Both are different.
They're both different, right? They have different slopes because the lines are not parallel, and they cross the y-axis at different points. Where they cross each other is irrelevant, right? Whether it's above, on, or below the x-axis is irrelevant. I'm calling all of those non-competitive. Okay? So in the first case, if you draw a little chart for me, right? So I want you to I usually draw this on the board when we do this in class, in the classroom, but we're doing it online. But make a little chart for me. You can generate it yourself. I want you to write out 4 conditions. We're going to talk about a pair of lines, right? So condition 1, the lines have the same intercept but different slopes. So write that out. Same intercept, different slopes. Condition number 2, a pair of lines have the different intercepts but same slope. So write that out. And then the third scenario, the pair of lines have different slopes and different intercepts. Okay. Is there a fourth scenario? Looking at what you just wrote, isn't there a fourth option?
Same intercept, same slope.
And what would that be? You're absolutely right. What would that represent?
Just not inhibited at all.
That would be the same line, which means not an inhibitor. So we can't really put that in a category, can we? That's not an inhibitor at all. This is why there are 3 classes of reversible inhibitors. Right? There isn't a fourth one because the fourth one is a default, not an inhibitor. It's, defunct case. So this is why we have 3 cases. So in the first case at the top, competitive inhibitor means same intercept, different slopes. Uncompetitive means same slope, different intercept. And in the latter case at the bottom, different slope, different intercept. Okay, keep those in mind real quick. Let's go back to our schemes on the right. We're going to put it together for you. I want you to compare what can competitive at the top, or what do competitive at the top, we're looking at the schemes on the right, not the graphs anymore. What does competitive at the top and non-competitive at the bottom have in common that the second one, uncompetitive, most certainly does not?
Different slopes.
We're not looking at the graphs. We're looking at the schemes on the right. Right? All these schemes over here on the right with the ES and E plus S and so forth. What does the competitive scheme at the top and the non-competitive scheme at the bottom have in common that most certainly excludes the uncompetitive scheme in the middle?
The inhibitor interacting with the free enzyme.
Exactly. The inhibitor can bind to free enzyme in both competitive and non-competitive. Agree? And uncompetitive is not in that club. Okay, so that's what competitive and non-competitive have in common, the ability to bind to free enzyme. And uncompetitive is not in that club. Move over to the graphs. What do the graph at the top and the bottom have in common, but the middle one definitely does not?
Different slopes.
A changing slope, exactly. So what does that imply?
changing slope is related to the inhibitor binding to the.
Binding to the free enzyme, right? So let's say it this way. The ability to bind a free enzyme does what?
Alters the slope.
Changes the slope.
Changes or alters the slope, right? So the ability to bind a free enzyme changes the slope. That's a positive thing causing a positive thing. What about the inability to bind a free enzyme? What can you tell me about that one?
Going to
Changes the y-intercept.
No, that's, you're drawing conclusions that aren't true. The inability to bind a free enzyme would be the inability to change the slope. It's just a corollary to the other one, right? Just because something can't bind to E doesn't mean it changes the intercept, because a no inhibitor can't bind to E. So the ability to bind a free enzyme changes the slope, and the corollary to that is the inability to bind a free enzyme is the inability to change the slope. Those are the same statements. Okay. Now I want you to compare two others for me. Compare uncompetitive in the middle, the schemes on the right we're talking about, uncompetitive in the middle with non-competitive at the bottom. What do they have in common that definitely competitive is not a member of that club? So what does the middle scheme and the bottom scheme have in common that competitive at the top does not?
interacts with the ES complex.
Right, so the ability to bind to ES complex. And then do the same thing on the left with the graphs. What does the middle graph and the lower graph have in common that the first one doesn't have at all?
Different y-intercept.
A changing y-intercept, okay? So draw the conclusion.
The ability to interact with the ES complex will.
So the ability to bind to ES complex changes the y-intercept. And the inability to bind to ES complex would not change the y-intercept, or has the inability to change the y-intercept. Right, do we see the two sentences now? Right, it's two sentences and their corollaries. So the ability to bind to free enzyme changes the slope, and the ability to bind to ES changes the y-intercept. Now look at those K's I put on there, KIS and KII. The K stands for constant because German starts with a K. And then the first I stands for inhibitory, so it's an inhibitory constant. That makes sense. But I don't just call it KI like a lot of the books do. It's called KIS and KII. And what does the S and I stand for? The S stands for slope and the I stands for intercept, meaning y-intercept. So KIS is the ability to change the slope. It's the inhibitor that changes the slope. KII is related to the inhibitor that changes the intercept. And for a non-competitive case, we need both numbers because both the slope and the intercept can change. That's what those numbers stand for. Okay? Right. So on to this one, and I'm going to leave the answers on the screen to save a little time here, but I'm asking you another question. Looking at competitive here again, at the top, the one in red. If I ask you what its KIS is, you'll give me a number. It's the rate at which EI falls apart. Right? Very simple. Let me leave it on here to make it simple for you. Look at the top. No other complications here. So KIS is the rate at which EI falls apart into E plus I. If I binds to the enzyme, if the inhibitor I binds to the E free enzyme very, very tightly, what do you expect KIS to be?
Small.
A very small number. It has the same relationship to affinity that Km does. You're exactly right. If the inhibitor binds to the enzyme, I binding to E, very, very tightly, then it doesn't fall apart very often, does it? And thus the rate at which it falls apart, KIS, will be small. So a small Ki or KIS means it binds tightly. What about for the middle one, the uncompetitive? If I binds to ES very weakly, what would KII be?
Large number.
A large number, because a way to say that I binds to ES weakly is to say that ESI falls apart readily. It's just another way of saying it. We like to say things as dissociation constants because they're so much easier to measure. Okay? So you see the relationship there. So what if I asked you, what is the KII for a competitive inhibitor? That would be right here on the graph. Under the ES in the first scheme, there's nothing written there. What would the KII be for a competitive inhibitor?
0?
Everyone always says that, so I'm not complaining, but it's not 0. What would 0
Does it just not exist?
Oh, well, it does exist, but it's not 0. What would 0 represent? KII represents the rate at which
That it would never fall apart?
that it would never fall apart. And that's not true at all. If ES, for a competitive inhibitor, if I were to try to bind to it, which we know it can't, but if it were somehow able to get on there, would it fall apart? Think about since I can never bind to ES in the competitive case, would the ESI, theoretically, if it were made, fall apart?
Yes, it would.
Yeah.
How well would it fall apart?
Like, perfectly.
Infinitely well, right? So a way of saying something never binds, since we always discuss it in terms of falling apart, as dissociation constants, so the KII for a competitive inhibitor would be what value?
Infinity.
Infinity. It falls apart infinitely well, is another way of saying it never binds. Since we're always talking about falling apart here. So that's why I wrote on the right, KII is equal to infinity for competitive. For uncompetitive, KII is a real number, but KIS is infinity because it never binds to E, or it falls apart infinitely well there. For the case of non-competitive, they're both real numbers. Okay? And for the special case of non-competitive that they like to call non-competitive in all the books, was where KIS equals KII, and that's very, very rare. Usually it prefers one or the other. Okay? And there's our equation on the left, is our normal Michaelis-Menten equation. And if we throw in these inhibitors, the KISs and KIIs, we get this arrangement where we're going to modify the constants Vmax and Km. We multiply them times these fractions. And it's how KIS and KII play into here. I want you to play with the equation a little bit, plug in those infinities and see what happens. And you should get back what happens to your Vmaxes and Kms that we talked about several slides ago, just using Le Chatelier's principle. Here's the mathematical rigor that backs it up. Okay, so play with those and make sure you come out with the same answers that we got just using intuition and Le Chatelier's principle to get to our answers. Okay. Very quickly, we'll talk about irreversible. Irreversible is where it binds and never comes off. It's kind of boring. An example of that at the bottom is penicillin. Right? It binds to a serines hydroxy side chain, makes a covalent bond to it. The enzyme can't remove it, and the enzyme is toast. It can never function again. Okay, so I'll let you read over. I'm not going to read the slide to you, but they're kind of boring, and once they bind, they never come off. They're useful drugs, but they're not as dynamic as the reversible ones in the analysis here. Okay. So quickly, let me tell you my analogy for this. It's my favorite one. It only takes a few minutes. But I'm going to need some participation from you guys. You got to contribute a little bit. Okay, so the schemes are on the right again. And we're going to talk about this dog named Watson. Kimberly named him for me as Sherlock Holmes' sidekick, Watson. He's a very smart dog, and he wanted to go to the high school prom. Who wouldn't want to go with him, right? He's got a rose and everything. So he wanted to go to prom. Of course, they told him, no, you're a dog, you can't go. So he goes anyway, and he brings his notebook with him. And they won't let him in the gym, so he decides to sit outside the gymnasium and look at the activities through the window. I know it's a sad picture. So he's looking at the activities through the window, and he's writing down what's going on in his notebook. He's going to publish this one day and win a prize. So he doesn't understand human behavior. He doesn't know what music is. So the analogy here is we are the dog. We can't see the enzymes and the substrates and the inhibitor molecules interacting. They're far too small. We can only observe what happens in the bulk, right? The result of many, many rounds of this happening, we see the outward effects of it. We can never see the details. They're too small. It happens too quickly for our human scales. The analogy here is the dog doesn't understand human behavior. He doesn't know what dancing is. He doesn't know what music is. He just knows that sometimes two of these humans are together while this noise is playing, that he doesn't understand music, and sometimes they're not together. They're not dancing, but they're not next to one another anymore. And he can visualize that. He can write down the time they spend together and the time they spend apart. He's got his Rolex watch and everything. Right? He's a very wealthy dog. Right? So he measures how much time you spend together and apart. So in this analogy, you and your date at the prom that the dog is watching are the enzyme and the substrate. Right? Which one's which, it doesn't really matter. But you and your date, E and S, spend some time together as an ES complex while the music's going. And when the music stops, you separate into E plus S. We'll leave the E plus P out of it. You can make up your own, not PG-13 analogies for yourself. We're only going to talk about the binding part, E plus S and ES. Okay, so what happens here, we have to make some assumptions that are true about enzymes and not true necessarily about humans. E and S have a certain affinity for one another that will never change. Right? They bind each other with a certain affinity under standard conditions and so forth, and that will never change. They have a certain number, right? They have a Km, right? You and your date, when you arrive at prom, may not necessarily have the same affinity for one another throughout the night, right? So we got to leave that human aspect out of it. We got to be true enzymes where the affinity for one another, the true affinity for one another, will never change. So E and S have a certain affinity. So what happens? Well, the dog sees you dancing. It sees you not dancing. It sees you dancing. It sees you not dancing. He writes down all these times. He calculates the time spent together versus not together. You have a certain affinity for one another. Wonderful. And then as he's watching through the window, he notices you being the substrate, right, walk off and go get some drinks for you and your date. Right? You go get some drinks at the bar. And you're over there. You're going to be a while. There's a line. You're coming back. And I need a name from the audience here. Somebody give me a name of a classmate that you had in high school that's preferably not in your class right now. That was kind of a competition for you, right? Like an adversary. Just any name will do.
Charlie.
Okay, Charlie. So Charlie's our first inhibitor. While you're off getting drinks, Charlie comes along and dances with your date while the music's playing. Right, so Charlie is binding to your date, forming an EI complex, right? And the dog writes that down, that they spend a certain time together. Then you come back with the drinks, right? You set them down at your table, and you want to dance with your date because the music's playing, but you can't because Charlie's dancing with your date. And you're not going to cut in. Enzymes don't do that, or substrates don't do that. So you're just going to wait until they finish their dance. The song ends, they separate, and go away. And then the next song, you dance with your date again. From the dog's point of view, you are spending effectively less time with your date. Does that make sense? The true affinity between you and your date has not changed. There's no animosity or anything. It doesn't change. But effectively, you've spent less time with your date. So the apparent affinity that the dog's calculating based on the time you spend with one another has gone down. So the effective or the apparent Km has gone up. What kind of inhibitor is Charlie? Charlie will dance with your date, but never you, of course. And while you're dancing with your date, Charlie doesn't interfere, of course.
He's a competitive.
He's a competitive inhibitor. It's a true competition. Your date either dances with you or Charlie. Right? It's one or the other. It's competition. It's the competitive inhibitor. It's a classic case. Okay? All right, I need one more name. Let's say someone who was considered like the class clown. Not Charlie. Let's pick a different name.
Teddy.
Who is it?
Teddy.
I still didn't quite understand the first part of it. Something Eddie.
Yeah, Teddy.
Oh, just Eddie. Okay, let's do Eddie. I thought there may have been another letter there. So Eddie's kind of the class clown. And Eddie will only dance with couples already dancing. That's why he's a clown. Right? So he doesn't dance with anyone individually. He will only run up to couples who are already dancing and bear hug them. Right? That's it. That's all he does. Whether the music's playing or not, he doesn't care. If you're dancing, he will come along and bear hug you. right? And have a weird conversation there holding you while you're dancing. And so when the music stops, you're supposed to separate from your date, and you can't because Eddie's just ongoing with this conversation, right? He's bear hugging you and your date. From the dog's point of view, it looks like you and your date are spending more time together than you normally would if Eddie wasn't around, right? So the apparent affinity between you and your date, from the dog's point of view, appears to be stronger. You appear to spend more time together. So it has an apparent lower Km. What kind of inhibitor is Eddie?
Uncompetitive.
Uncompetitive. This is all Eddie does. Eddie just showed up by himself and he only bear hugs couples dancing. Okay? Starting to make sense with the two so far? Remember, the dog's just putting it in his terms. Okay? So the next thing, I need one last name from the crowd. Not Charlie, not Eddie, I need something else. Another class clown.
Brandon.
Brandon.
Jack.
Okay, let's do Brandon. So Charlie was the competitive, Eddie's going to be uncompetitive, and of course Brandon will be our non-competitive case. So Brandon does like Eddie does. He likes the bear hug couples dancing, but he will also dance with your date if you're not around, right? It's very likely that Brandon has a preference for doing one or the other. It's highly unlikely that he does them in equal preference. So it's not going to be what they call true or perfect non-competitive. It'll be a little shifted one way or the other. From the dog's point of view, though, depending on Brandon's preference, you could appear to spend more time with your date and thus have a lower KM, like with Eddie. Or if Brandon prefers to dance with your date while you're not around over bear hugging, you could appear to have a weaker affinity with your date, right? And so from the dog's point of view, the KM could appear to go up, it could appear to go down, it could rarely look the same. And in fact, while Brandon's dancing with your date, you could show up and start dancing with them. Or while you're dancing, Brandon could show up and bear hug and you leave. That's what the bottom of that scheme is showed. So there's lots of possibilities, but the KM could go up, appear to go up, could appear to go down, could appear to stay the same. So does this, from the dog's point of view analogy, give us like our insight into how enzymes work? We can only see the outward gross accumulative effects. Hopefully this analogy helps put those together. Right. And there's one more part to the analogy and then we're done with it. Do you remember the drinks that you went to go get while your date was dancing with Charlie? Well, the dog noticed that too. And he's still kind of upset that he didn't get to go to the prom because he seems like it's kind of fun to run around the dance floor. He's still a dog. So he calls the police. He has a cell phone too, by the way. So he calls the police, and of course you shouldn't have had those drinks at a high school prom, so the police show up and they start arresting everyone for underage drinking. And what does the dog notice? From his point of view, everyone who's getting handcuffs on them is no longer dancing, even if the music goes on. So what kind of inhibitor are the handcuffs?
Irreversible?
They're irreversible. They tend to not come off, right? It's like a covalent bond. So from a dog's point of view, the handcuffs represent an irreversible inhibitor, right? So it's just a final thought on that analogy to try to put all those together for you. Okay. All right. So I'll stop there, and we'll continue with chymotrypsin next time.